Hello everyone, welcome to ShailajaIAS and welcome to the PYQ Decoder course. I am Subhash. I am continuing with the class 3 of Science and Technology for the UPSC Prelims 2026. Today, one of the important theme which we will discuss is biotechnology and health science. Massive number of questions will show up in these themes with respect to health, with respect to new initiatives in the health, any new treatments, with respect to basic biology, biotechnology related aspects, health aspects. All these questions have shown up with respect to biotechnology, health sciences and the allied applications of it. So we'll see through last 6 years ka PYQs. Chalo, we'll begin. 2025 में with respect to biotechnology and health, with reference to monoclonal antibodies often mentioned in the news, current affairs. Which of the following statements are correct? What are monoclonal antibodies? These are synthetic lab-made antibodies that mimic the natural antibodies present in our system, in our immunity. They are man-made proteins. Yes, correct. They are proteins, antibodies, and they are man-made, synthetic. They stimulate immunological function due to their ability to bind to specific antigen. This is the function of antibody. Antibody, what it will do? It will bind to the antigen of the pathogen and it stops further growing of the pathogen. It might be virus, bacteria. It will stimulate that. How naturally immunological function, how it functions, it will stimulate that and it will bind to the antigen of the cell which is affected or the pathogen itself. So both two statements are correct. They are used in treating viral infections like that of Nipah virus. Yes, they can be used in various initiatives like for treating cancerous cells, for treating infections due to bacteria, virus, all this can be true. And all the three pairs are correct. Here if you see 1, 2 and 3 are, three pairs are correct. So monoclonal antibodies, they are designated as MABs. And they are lab-produced molecules engineered to serve as substitute for antibodies. Okay, in body, generally you will generate antibodies for any pathogen. But if you are very sick and if you need a sudden assistance, what you can do? You can push antibodies into your bloodstream from outside, which are man-made proteins, to fight against the pathogens inside the body. So all the three pairs are correct here. Statement 3, MAB, specifically M102.4 have been used, okay, it was in news also for the treatment of Nipah virus infections. So it was used, it was in news that year, 24-25 Nipah outbreak also had happened. So based on that, question had come in health science, biotechnology and health science. Next, 2025. This kind of questions do repeat in UPSC every year. Okay, these are sure short questions. You have to answer them very easily. You will get 2 marks. No virus can survive in ocean water. Okay, UPSC will not check whether no virus will survive or not, even if one virus survives, and there will be always exception to a general statement. So this statement is wrong. Even a single virus survive in ocean water, it is wrong, this statement. No virus can infect bacteria. You know, entire class of viruses under bacteriophages, right? Bacteriophages. They are used as molecular scissors, right? DNA scissors, molecular scissors. In CRISPR, Cas9 technology, in that bacteriophages are used as molecular scissors, right? So you know this statement also is wrong. No virus can change the cellular transcriptional activities in host cells. This is also wrong. You have RNA viruses, right? Like for example, HIV virus will do reverse transcription. From RNA, it will generate the DNA. So RNA viruses. So all these three statements are wrong. Consider the following, which are correct he is asking. Do read. Sometimes which are not correct, if he has given, all three will be not correct. Here is given which are correct, none. Answer is None. This kind of statements will be there. Some viruses can live in ocean, some viruses can change. Instead of no virus, if some is there in all the three statements, all the three statements will be correct. That kind of question every year UPSC is giving you, offering you in a plate to get 4 marks, 6 marks. So please make that statements, all the three statements are correct. Exceptions do exist in all the classes of virus, bacteria, microorganisms, animal kingdom, plant kingdom. So don't take that, no virus can change cellular, except you know for a fact, knowledge. For example, virus need a host to survive. Virus cannot grow outside the host body. For example, virus only if it enters through body, it will grow and it will expand. It will expand inside the host body. But if it is outside, no, it cannot grow. It needs a medium, living host medium to grow. That knowledge if you have, you can answer. Otherwise, if this kind of generic statements, no virus can survive in ocean water, no virus can survive in acidic condition, even if one virus has adapted and it survives, that statements will be proven wrong. So make sure in this kind of statement you go with all. Here all of these statements are wrong. If you give some virus can survive, some virus can infect, some virus can change cellular, all statements will be right. Not all viruses will do that, but some viruses will definitely do all this. So here, none of the above answer. Next, again, 2024 may we'll see which one of the following is synthesized in human body that dilates the blood vessels and increase blood flow. If you have studied biology, 11th and 12th biology, that to some chapters with respect to biotechnology, health science, human body, and some of the concepts of human reproduction, biotechnology, how gene transfers take place. Questions have revolved in UPSC with surrounding that. Now, this is vasodilators. One of the options acts as a vasodilators to dilate the blood flow in the in your nervous system, in your blood system, in your blood streams. Which of the following options? Nitric oxide, nitrous oxide, nitrogen dioxide, nitrogen pentoxide. See. If you don't know the answer also, you can arrive at the answer. How? Just you have to keep an open mind. Sometimes in UPSC, whatever you have studied will come to the fore there. It will come, it will appear before you the answer. How I made it right in examination, in the prelims? See, nitrous oxide, I know it's a laughing gas. Laughing gas is not vasodilator, it will damage your body. Laughing gas is not used as vasodilator, that I know. Then nitrogen dioxide is a pollutant. Right? Nitrogen dioxide and sulfur dioxide are a pollutant, are main causative agents for acid rain. Pentoxide, the higher the oxygen level, higher is the toxicity, that I know. So nitrogen dioxide, nitrogen pentoxide cannot be the answer. These two definitely I'll rule it out. And I know nitrous oxide I studied in 11th standard chemistry. Nitrous oxide is a laughing gas, which we will use it in say helium balloon, or nitrous oxide can be used in medical other appliances, but it is not generally used for vasodilation. So if I eliminate these three statements based on my knowledge which I have studied just in 11th and 12th, so I will get nitric oxide. You should study. If you are not studied, then you will not be able to answer. But if you are a biology student, or if you are a PCM student, also chemistry if you have studied, and if you know normally nitrogen dioxide and higher oxides of nitrogen are pollutants, then at least you can end up with 50-50. That is the benefit of having an open mind and just seeing through options and arriving at the answer. So here you will arrive at answer nitric oxide. Nitric oxide or NO. This is N2O, nitrous oxide. A chemical change can make it very dangerous to human body. NO acts as a signaling molecule in the cardiovascular system. It signals the smooth muscle of the blood vessels to relax, vasodilate, thereby increasing blood flow and lowering the BP. If there is a high BP, to reduce the BP and to relax the muscles and increase the blood flow, what you will use? Nitric oxide. So first option is correct here, and you can eliminate other options just by the knowledge you have possessed till what you have studied in 10th and 12th physics, chemistry. If you have studied chemistry and biology, you can answer. Without that also, you know, two oxides, nitrogen dioxide and pentoxides are pollutants, then you will end up with 50-50. If you know nitrous oxide is a laughing gas, then you can end up with the first option. Clear? Next question. In which of the following are hydrogels used? Control drug delivery in patients, hydrogels, mobile air conditioner systems, preparation of industrial lubricants. Unless, sir, you know for a knowledge these are not used in this specific appliances, go with all of the above. Of course, in 5 questions, 1 or 2 questions, UPSC might give wrong. Not all of the above will be the answer. But that you have to go with the knowledge and understanding of the concepts of where specific, chemical specific objects are used in which applications, which materials have which applications. This you need to make a table. Which important materials that are in use. In Hindu, the scientific editorial every Sunday when it comes, these kind of hydrogels, nano microsatellites, all these are discussed and their applications are discussed. If you can't do it now anyway, just you can do which are the important scientific discoveries, scientific research going on, and new items like this, what are its applications, just make a list. If you have it, most of the questions will be all of the above. However, UPSC will give 1 or 2 questions as wrong. Here you can see all these 3, 1, 2, 3 hydrogels are used. Hydrogels are 3D network of hydrophilic polymers that can hold large amount of water. They are hydrophilic. Philic means attracts the water. They are widely used in medicine and drug delivery, wound dressing, in agriculture for soil moisture, and in various industrial applications due to unique physical properties of hydrogels. So here all of the above is the answer. Membrane bioreactors. I think we did it in the environment section. Membrane bioreactors. I told membrane normally to filter. So if you see assisted reproductive technologies, you need to know, options become important. Drug delivery nanotechnology, you need to know. Vaccine production technologies, various methods of vaccines are produced, mRNA, adeno, vector-based vaccine. So you have to know different types of all this can be a potential question and has been asked in UPSC. Wastewater treatment technology. This is the answer. Why? Membrane. Normally in water filtration also reverse osmosis. Membrane bioreactors also act as a wastewater treatment technologies. I think we did it in environment. So here answer is D. So now we'll see 2023 questions. See again. Some mushrooms have medicinal properties, some mushrooms have psychoactive properties, some mushrooms have insecticidal properties, some mushrooms have bioluminescent, like glowing properties, bioluminescent properties. Which of the following statements are correct, sir? All. I'd close my eyes and I'll put all 4. Every year or once, 1 or 2 questions, UPSC is repeating this kind of questions. It's offering in your platter, make all the questions right, sir. Here you take 4 marks easily. So this is where you have to not overthink and take it right. Even if one fungus does this, even if you don't know, all the statements are correct. Just like no viruses, none of them were right. Same thing here, some, all will be right, because if one mushroom also does this, have these properties and characteristics, exceptions will always be there and will have medicinal properties. Some mushrooms can have medicinal properties, some fungi can have, So mushroom is a fungi class, right? Fungi. So all these statements are correct. Next question. Wolbachia method. That year it was in news. Wolbachia method is sometimes talked about with reference to which of the following? Controlling the viral diseases spread by mosquitoes, converting crop residues into packing materials. That was also in news, E-Pusa. To make it a packing material and making it biodegradable, all this. Producing biodegradable plastic, producing biochar from thermochemical conversion of biomass. All these are some of the technologies of conversion to energy. So here if you see, Wolbachia method was mainly to control mosquito population, Aedes aegypti to control chikungunya, okay, malaria, Zika virus, dengue. In order to control Aedes growth and spread of Aedes mosquitoes, Wolbachia method was used. So here answer is option A, and that year this technology or this method was used. It's a bacteria which was introduced into Aedes aegypti mosquitoes, and it competes with viruses like dengue, Zika, chikungunya, and making it harder for mosquitoes to transmit this disease. Okay, these diseases. Next, aerial metagenomics best refers to which of the following situations? As I told you in economy and any this one, whenever it gives term, you just decode it. Aerial, okay, aerial, okay. Drone was aerial, it was airborne. So airborne, something that is in air, airborne. Metagenomics. Metagenomics. Genomics is related to gene, DNA. So now take this and answer. Collecting DNA samples from air in habitat at one go. Yes, DNA, air. Okay, this is matching. Understanding the genetic makeup of aviation species of a habitat. That is not called aerial metagenomics. In air collecting DNA, not aerial. Aviation species ka connecting DNA genetic makeup, okay, but it is not directly mentioning aerial metagenomics. Using airborne devices to collect blood samples, not blood samples here, nothing with respect to blood samples, okay, it is metagenome, DNAs. Sending drones to inaccessible areas to collect plant and animal samples from land surface and water bodies. This is also not directly referring. If you see A and B, you might be confused, but this is aviation species, airs, right? You have different species, pieces, fishes, airs, okay? So this will be also not an answer, but if you break down the term, you can see aerial metagenomics. Aerial, it is airborne, collecting DNA that is airborne in a habitat at one go makes very sense, and you can eliminate other three statements here. Answer 1. What it involves? Studying genetic material recovered directly from environmental samples in the air, and aerial metagenomic specifically targets eDNA or environmental DNA suspended in the air to monitor biodiversity. Right? You no need to capture any animals, birds, just the DNA left with the flora and fauna in the air can be used to check and measure the environmental biodiversity. Next, microsatellite DNA. Microsatellite DNA is used in the case of which of the following? Studying the evolutionary relationship among various species of fauna, stimulating stem cells to transform into diverse functional tissues. Okay, this is totipotency, pluripotency, right? Converting the stem cells into other cells, other tissues, including making entire, okay, person itself, entire body you can create. That is totipotent. Promoting clonal propagation of horticultural plants, again cloning, but that is not related with microsatellite DNA. Assessing the efficacy of drug by conducting series of drug trial in a population. What is this called? Drug trials, okay, trials in a population. That is not, however, directly linking to microsatellite DNA. If you see, two statement B, C, D are not at all defining or pointing towards microsatellite DNA. However, if you see, microsatellite DNA is studying the evolutionary relationship among various species of fauna, animals. That is how species has evolved. And subspeciation. Okay. From one species, another species will get created due to many factors, due to mutation, due to environmental factor, due to natural selection. Okay, all this you have studied in Darwin theory, all that. So microsatellite DNA are repeating. Okay, we know adenine, thymine, guanine, cytosine is there in our DNA, ATGC. Some of this DNA proteins will repeat. For example, AT like this, it will repeat. These are repeating sequences. These repeating sequences are called genetic markers, or also called short tandem repeats or genetic repetitive DNA markers, and they highly mutate. Just by scientists seeing this, they can know how the evolutionary relationship has been formed from one species to other species and where subspeciation has been formed. It was in news and it's a core concept which we study in genomics, okay, and genetics. So here answer A. Next, consider the following statements from 2022. Biofilms can form on medical implants within human tissues. Okay, biofilms are microbes. It can be beneficial microbes which are in our own body. If we have some implant in human tissue, biofilms can form on that. Yes, it can form on food and food processing surfaces. Yes, microbes can form a biofilm. Biofilm can exhibit antibiotic resistance. Possibility, sir. Yes, it can exhibit antibiotic resistance. So your answer will be 1, 2, 3. Again, close your eyes and do. Unless you know for a fact or you have knowledge that this can't be done, please go with all of the above. Even if 1 or 2 question is wrong, maximum number of questions will be right. If 4 or 5 questions come like this across subjects, and if you attempt 5 questions, if 1 or 2 wrong also, you will be net positive. Okay, 3, 4 minimum will be wrong. 1 might be an exception sometimes, but that will be a concept based, knowledge based question that you need to have clarity on the basic concepts of how things work. So these are the clusters of microorganisms that stick to surface and they can have all these characteristics. Next, with respect to probiotics. Okay, probiotics are the beneficial bacteria like lactobacillus. It will help in digestion of milk, lactose. Probiotics are made up of both bacteria and yeast. Yes, the organism in probiotics are found in foods we ingest, but they do not naturally occur in our gut. As we are born only, some bacteria are there which are naturally occurring in gut. Right, this is the concept. Only occurs outside but not naturally in our gut. Okay, always a trap of UPSC, famous trap. It will occur in that, but when he uses but do not, he will keep a trap and he will make that statement wrong. It can help in digestion of milk sugars. Yes, we saw lactobacillus, yeast, Saccharomyces. So here both 1 and 3 are right. However, 2 is a trap, and whenever he is using but, he is keeping a trap. So answer is 1, 3. So option C. Clear? I hope this question is clear. Now we will go to next question with respect to COVID. Okay, that time COVID was there. So COVID question had come. We'll see. In the context of vaccine manufactured to prevent COVID-19, consider the following statements. Sir, anything with respect to vaccines, with respect to new drugs to treat cancer, to treat HIV, to treat aging, to treat new diseases like Alzheimer's, some autoimmune diseases, and any drugs that has been removed from patent and generalized. India is a factory. It's a factory to produce the generic drugs of the world. So you need to have idea and you need to have new current affairs things going on this thing. So Serum Institute of India produced COVID-19 vaccine named Covishield using mRNA platform. No, it was wrong. Like Pfizer. Okay, Pfizer produced using mRNA technology. Covishield used a viral vector, AstraZeneca, not mRNA, right? Then Sputnik 5 vaccine is manufactured using vector-based platform. Okay, Russian-based company did it. Yes. Covaxin is an inactivated pathogen-based vaccine. That is also correct. Bharat Biotech used this and it killed or inactivated virus. Okay, half defeated or killed virus, it injected in our body so that immunity will be there when the actual COVID virus, coronavirus will attack the human body. Here if you see, statement 1 is wrong and statement 2 and 3 were correct. Okay, however, this was in 22. So now this question might not be repeated again, but you should know which new initiatives and which questions can be asked in 26. Right, sir. Which one of the following describes the role of B cells and T cells? Okay, these are again cells that are involved in immunity and disease prevention. B cells and T cells. So they protect the body from environmental allergens. They elevate the body's pain and inflammation. They are like eosinophils, neutrophils in our body. They act as immunosuppressants in the body. They protect body from disease caused by pathogens. They directly attack the pathogens. B cells and T cells are the primary components of the adaptive immunity system, where B cells produce antibodies, which we studied in the first to neutralize pathogens. They will attach to the antigen of the pathogen, while T cells either directly direct the immunoresponse or directly kill the infected cells. Okay, it will give an alarm to other immunity system to get activated, and also it will directly kill the cells which are already infected by pathogens. So B cells and T cells protect the body from the diseases caused by microbes and pathogens. So next question. DNA barcoding can do what? Assess the age of plant or animal. That is it DNA barcoding? No sir. Assessing the age of the plant and animal is not DNA barcoding. Distinguish among the species that look alike. DNA barcoding can differentiate between the species that might look same but they have different DNA. Identify undesirable animal or plant animal. Yes, if that DNA is found, then you can separate it from the undesirable animal and plant DNA. So here what you use, assessing the age of plant and animal, you use dating, carbon dating. Biodating also you can use, phosphorus dating, carbon dating, dating methods you use, but DNA barcoding can use to find the DNA in any given material. So distinguish among species, identify undesirable animal or plant DNA can be done. However, carbon dating, biodating can be used to assess the age of plant or animal, and that cannot be done with DNA barcoding. So clarity of concepts here, sir. So 2 pairs are right, 2 and 3, and 1 is wrong. Why? DNA barcoding is not used in calculating the age. That is done using the carbon. Example is carbon or phosphorus present in the plant or animal fossil. Next question. With reference to, it was in 2021 and news also, recent developments regarding recombinant vector vaccine, consider the following statement. Recombinant means you are recombining the DNA genes. Genetic engineering is applied in the development of these vaccines. Yes, correct. It was in news. Bacteria and viruses are used as vectors. Yes, both bacteria and viruses were used as agents to transfer these genes. Here, both the statements are right and straightforward question this is. So, used in genetic engineering to insert genes, DNA, from a pathogen into harmless vector. It can be virus like bacteriophage and different bacteria, and this will carry the gene into our body cells to trigger an immunoresponse. Example include AstraZeneca, Covishield vaccine. It used adenovirus as a vector, and you can have many bacterial vectors also. So here both 1 and 2 were correct, my friends. Next, again, in the context of hereditary disease. This is core concept. Consider the following statement. Passing on mitochondrial diseases from parents to child can be prevented by mitochondrial replacement therapy either before or after in vitro fertilization of the egg. This is correct. It was in news also. So, Pronuclear transfer technology. Normally, any diseases with respect to mitochondria is what? Powerhouse of the cell, and the baby normally will get the mitochondria from female. The baby will always get mitochondrial DNA, a small DNA in mitochondria from only female, and it can be prevented from changing or preventing the mitochondria of mother to be removed and by replacing the healthy mitochondria. So it is also called three-parent baby, removing the mitochondria from the mother that is coming to baby, and you are taking the donor mitochondria and putting it in the baby, healthy mitochondria. So it's also called three-parent baby. It was in news that year. A child inherits mitochondrial diseases entirely from mother and not from father. As I told you, with respect to this concept, you know mitochondria is directly coming from mother and not father. So your answer was both 1 and 2 asked in 2021, 2020. Before that also it was asked 3 years continuously. This theme was repeated. Likewise, in any new research with respect to health, with respect to like this, will be very important. In vitro fertilization, with respect to IVF, with respect to diseases, immunity diseases, with respect to hereditary diseases, it will be very important that you properly prepare, which is in news. Mitochondrial replacement therapy, MRT, often called three-parent baby technology, involve replacing faulty mitochondria in a mother's egg with a healthy one from a donor. This can be done via pronuclear transfer after fertilization or maternal spindle transfer before fertilization of the egg only, because mitochondria are located in the cytoplasm of the egg, not in the sperm. Not in the sperm. Therefore, it is inherited only from mother and not father. Clear? 21 May. Bolgard 1 and Bolgard 2 technologies are mentioned in the context of clonal propagation of croplands, developing genetically modified croplands, production of plant growth substances, production of biofertilizers. This was used in BT cotton. So, GM croplands. Bolgard 1 and Bolgard 2 are specific biotechnological brands of BT cotton. Bacillus thuringiensis is a bacteria. It's a GM cotton. Only approved crop in India which is grown is GM cotton. It involves insertion of this gene Cry1AC and Cry2AB from the bacteria Bacillus thuringiensis, so that it is resistant against the bollworm pest. So it will not eat the pest will not eat the cotton leaves through if you grow BT cotton, which includes Bolgard 1 and Bolgard 2 genes, which was resistant to bollworm, the pests. So it is option B, developing GM crops. So which will control GM crops in India? GEAC, right? Under Ministry of Environment, Forest and Climate Change. Now, consider the following: bacteria, fungi, virus. Which above can be cultivated in artificial synthetic medium? As I told you, except virus, all can be grown. Why? Virus needs a living host to multiply. So, except virus, bacteria and fungi can be cultivated in artificial synthetic medium. This is proper basic science biology which you have studied. If you apply, only 1 and 2 and not all of the above here. Not all of the above. Why? Because virus can only grow in the host cells and it cannot be artificially just by cultured in a synthetic medium, but just by providing nutrients and food for the bacteria or fungi. How you give food? You cannot just give outside food for the viruses. It needs living host to grow. Some RNA viruses need, because it needs the host DNA to reverse transcript and generate RNA copies. So your answer was 1 and 2 only. Next question, sir. Adenoviruses have single-stranded DNA genomes, whereas retroviruses have double-stranded DNA. Retro means they reverse the their RNA to form, reverse the DNA of the host cell to form their RNA. So here he has changed. Retroviruses have RNA, no DNA, and adenoviruses normally have double-stranded DNA. Statement 1 is wrong. Common cold is sometimes caused by adenovirus, whereas acquired immunodeficiency syndrome, AIDS, is caused by a retrovirus. HIV, human immunovirus. Yes, this statement is correct. This is a RNA virus, and adenovirus causes the common cold. Here, only one pair or option 2 is correct. Again, easy. You can see also how easy it was in 2021 and how it has evolved across 22, 23, 24 and 25. Now, the term ACE2, which we saw already, right, in Bolgard, in BT Cotton, is talked in the context of genes introduced in genetically modified plants, development of India's own satellite navigation system, radio collars of wildlife tracking, spread of viral diseases. Okay, ACE2 is angiotensin converting enzyme 2. Okay, it's a protein on the surface of many cell types, and it gained prominence during COVID. Why? The pandemic, because it acts as an entry point for the COVID virus to infect human cells. So, spread of viral diseases, ACE2, that was in news in that year. So, question had come directly from there. So, this might not get repeated, but just to see how the question has, how the difficulty of the question, okay, the language of the question has transferred from 2020 to 25, you'll be able to see. The context of recent advancement in human reproductive technology, pronuclear transfer asked in 20 and complete detail was asked in 22. It is preventing mitochondrial diseases in offspring. Direct, easy question, and its conceptual understanding is asked in 22. So before that also, question on mitochondria, okay, pronuclear technology was asked. Now, genetic changes can be introduced in the cell that produces egg or sperm of a prospective plant. Yes, you can do in gene change. A person's genome can be edited before birth at the early embryonic stage. Yes, it's possible. Human-induced pluripotent stem cells can be injected into embryo of a pig. Yes, that also you can do. Here, all three are possible using new biotechnological methods, and germline editing, this both are. Statement 3 is creation of chimeras, where human stem cells are integrated in animal embryos like pigs to potentially grow and do organ transplantation. So here all the three statements is correct and applications of biotechnology. With respect to pneumococcal conjugate vaccines, okay, very different types of vaccines are there. I'm sure you would have studied. With respect to this, you see these vaccines are effective against pneumonia as well as meningitis and sepsis. Dependence on antibiotics that are not effective against drug resistance bacteria can be reduced. Yes, generic advantage of vaccines. These vaccines have no side effects and cause no allergic reactions. Wrong. During COVID also and during other vaccines also, you see many side effects and reactions taking place. So 3 statement, third is wrong. If you eliminate third and you know this is statement 2 is right, you will be able to get 1 and 2 answer. Yes, they are not only effective against pneumonia, they are also effective against meningitis and sepsis. So statement 1 and 2 are correct here, and 3 is a wrong, and see the trap kept by UPSC. Very extreme statement. This is very basic of plant cell and animal cell. Plant cells have cellulose cell walls while animal cells do not. Yes, animal cell do not have that extra layer of cell wall, and plant cells do not have plasma membrane unlike animal cells which do. This statement is wrong. Both plant and animal cells have plasma membrane above which plant cells alone have cellulose walls. So statement 2 is wrong. Mature plant cells have large vacuole while animal cells have many small vacuoles. This is also basic characteristics and difference between plant and animal cells. So 1 and 3 is correct. Basic of plant cell, animal cell, and second statement is wrong. Both plants and animals have plasma membrane. So you need to know here basic in biology, plant cell, animal cells, different cell organelles, their functions. Okay, this biology if you cover, and if you cover current affairs, many of the question in this topic you can easily answer. This was a very exhaustive where 30 plus questions were there in the last 6 years. So every year 4 to 5 questions show up. In the last theme, IT, AI, emerging technologies, many questions are coming 4, 5, and here 3 to 4 questions minimum will come. So 70% of the questions will come in these 2 themes. And the next important theme is other technologies like defense, space, nuclear technology, material, energy. All these questions will be coming from these 5 micro themes under a larger umbrella. Okay, so in the next class we'll be discussing those questions. Now make sure you properly prepare and you align with self. Okay, where you align with these 2 important themes of science where you will get easily 8 to 9 questions. Till then, I'll see you in the next class. Take care.